Good morning, let's have a class. Some of the announcements I just want to mention. We will have today talk about Clostridium, some sexually transmitted disease. Then Thursday we finish about that section, talk about antibiotics. The next week there is no lecture because October 3rd is a lab exam, midterm exam. So we don't have a lecture that day, and October 5th is fall break. So go back. Hopefully October 10th we will be doing a review. We're going to do a review that day, and then October 12th will be the second exam, okay, of the lecture. So that will be the plan. Now here's something I want to mention. I already put on the e-campus. Some of the material for the after-class reading, you please read it. Because instead of have a textbook, I did lots of these conclusions, okay, for all these read. Some of the questions looks like this, case study exam review, that could be similar in the exam. And the same thing for this after-class reading too, you should read it. Okay, this is what we talk about, the Corynebacterial diphtheria. And basically is what I wrote on the blackboard is, I wrote it there. Now, I want to tell you one thing. If you guys think about it, you don't want to read anything. Okay? You're a little bit busy. Any reason? Then go to look at your lab menu. In your lab menu, I have been put it similar, which is a case study. Where it is, I need to find for you. Somewhere, okay, right here. If you don't want to read that tedious stuff, you could read chapter 19 of the lab manual. It's the same thing. It's a little bit concise compared to what I put there in the after-class reading. And you also could read, not this one. This is we'll do later. Chapter 21. So chapter 19 and the chapter 21 of your lab notebook, lab menu, also could be your study guideline, because this is what I wrote to myself, and it's close to what the lecture talks about. But I only encourage you just to read through the after-class reading material, okay, because that's more detailed and more important. If you don't really don't want to read anything, read the lab menu, chapter 19 and chapter 21. Okay, so let's talk about the lecture 2, exam 2, lecture 2. Okay, in this section, we want to talk about is a human digestive tract. So you could see the oral cavity is esophagus, liver, stomach, pancreas, large intestinal, an anus, rectum, appendix, small intestinal. So we want to mention several things. Is human digestive tract. Okay, there are several things we want to mention. Number one, stomach. The bacteria number is very low. It's lower than 10 to the second. So lower than 100 cells per ml. Why? You know the reason? Because stomach acid. Stomach acid is very low. The pH somehow is 1 to 2. So most of the bacteria will be died. So the number of the bacteria in the stomach is very low. In the small intestinal, it starts to increase the bacterial number. Small intestinal, somewhere about 10 to the 4th to 10 to the 5th. However, when they go to large intestinal, including your stool sample, okay, or we call it a fecal sample, that bacteria is huge. Could be up to 10 to the 12 cells. It's a huge. And I want to tell you one thing. At large intestinal, majority of them is anaerobic bacteria instead of aerobic bacteria. So the ratio is somehow about 1,000 to 1, even more than that sometimes. So majority of them is anaerobic bacteria. And our fecal sample, 1/3 is bacterial cells. So that is the reason there is a modern technology called 16S rRNA could do analysis of the intestine of the bacterial population, because there is a huge bacterial community there in a large intestine. So you get a fecal sample from the animals, you can see their profiles, their dynamics, and all kinds of information will tell you. And it also, there is a theory talk about our most important immune system is not B cell, is not T cell, is not spleen. It is actually our bacteria in the body. The bacterial community in the body is the largest and the most important bacterial immune system in our body. Okay, so that is something I want to mention. The whole human digestive tract, the bacterial situation. So that's the slides talk about. This slide, which tells you something, is another concept, which is food infection versus food intoxication. And we talked about this somehow before briefly. So first of all, food infection, we said very easy. You are eating bacteria. And the food intoxication, you are eating toxin. So what are the examples for the food infection? When we talk about foodborne pathogens, and the foodborne pathogen, when we talk about salmonella, and E. coli O157H7, and Listeria, those are food infections. In other words, the easiest way to prevent food infection is roughly cooking. So you cooking, you could solve the problem very easy. Because you are eating bacteria, when the bacteria died, the problem solved. Food intoxication, you are eating toxin. The example, Clostridium, and the bacillus, and Staphylococcus. And they generate, which is a toxin. That could be exotoxin, that could be enterotoxin, all kinds of toxins, and most of them are heat resistant. So, for food intoxication, you can't just simply use cooking methods to solve the problem, because the toxin is already there. And I also tell you, the onset time is very short. Could be only 6 hours. You will have vomiting, nausea, and the diarrhea. Very quick. That's exactly what do you find here in AES. Nausea and abdomen cramping, all those kinds of symptoms. Think about you eat something raw, and you have diarrhea, vomiting, nausea. Okay, so this is some concept I would love to mention. Beginning we talk about. Here they give you 3 examples, which is Staphylococcus aureus, Clostridium botulinum, Bacillus cereus. These are all cause food intoxication. And you see the onset time of the symptoms is very short, 1 to 6 hours, averagely about 6 hours. And we know Staphylococcus aureus is actually normal bacteria sometimes on the skin. And chicken salads is one of that. If you do sneezing, then you contaminated it, you have a diarrhea in like 6 hours. And very ironically, this pathogen is not required to report to CDC. So people think about you have a couple of diarrheas and you recover by yourself, even without using antibiotics. But Clostridium bacillus, those spore-forming bacteria, sometimes cause a problem. And the Bacillus cereus, most of the time, is existing in the rice. So if you have a rice, cooked the rice, even you left on your bench for a longer time, and about 3 weeks later, you see the little spore going to be forming. Okay, so that's something I want to mention. Now, today's class, we will spend lots of time talking about a group of the bacteria is Clostridium. So here we want to talk very detailed about the Clostridium, and basically it's 4 different types of the Clostridium. That will be the key of this section. And we won't do these in the lab because Clostridium is biosafety level 3, and we don't have that lab facility to do it. Okay, so Clostridium. I will just write Clostridium SPP, species. So first of all, Clostridium, overall, their gram-positive large anaerobic bacteria, and very important, endospore forming bacteria. So these two are the key. It is anaerobic bacteria, it is endospore forming bacteria. Now remember, anaerobic bacteria we talk about, aerobic bacteria need oxygen, anaerobic bacteria cannot survive in the presence of oxygen, and we have another group, which is most of the bacteria belonging to this group is called a facultative. And remember the question in the lecture exam, the extra points, why anaerobic bacteria cannot survive in the presence of oxygen, because there is no Das mutis, is that right? Sulfide, superoxidized Das mutis, and they also know catalysts. That means generate the toxin during the aerobic respiration. We need these enzymes to hydrolyze. However, this bacteria doesn't have that. So it cannot survive in the presence of oxygen. So that's anaerobic bacteria. And it's a strict anaerobic bacteria. In the tubes, you will see it grow at the bottom, usually at the precipitation. Okay, this is overall we want to talk about. Now, what are the species we want to talk about? Is the Clostridium, botulinum, botulinum, Clostridium tetanus, Clostridium perfringens, and the last one, Clostridium difficile. We will introduce all these in much more detail. Okay, first of all, Clostridium botulinum. Botulinum. Clostridium botulinum. Genus name, species name is already said it's botulinum, so it will cause a symptom called botulism. And there is basically 3 different types of the botulism, which is, first one, wound botulism. Second one, infant botulism. And the last one is food botulism. Okay, the first one is easy. What is wood botulism? You always remember, because Clostridium is an anaerobic bacteria. So whenever in your body, if there is a cut, something on your skin or there is a cut, and then you contaminate it with soil or some decayed metal, then the bacteria could start to survive and grow and cause infection. Why? Because in the cuts area, it is somehow microaerophilic to anaerobic. That is generally a condition for Clostridium to grow there. That's a wound botulism. And the wound botulism also very typical for drug abuser or even alcoholic person. It's a risk group. It's very interesting. That's a wound botulism. Said very simple. And you can see an example which looks like the picture 4 on the right side. Okay, that's what I want to introduce to you. Infant botulism, very easy to understand. Just use one sentence. Never feed less than 1 year old baby honey. Otherwise, you will have a floppy baby. It is a joke, but it will let you understand it very well. That's a floppy baby. Look at that. So, we want to talk about that. Why? Okay, where does the honey come from? Originally, honey comes from sugar. Where's the old resource? Summer soil. Okay, they could be contaminated with Clostridium, all kinds of Clostridium. Why the baby is less than 1 year old? Because the normal intestinal bacterial flora has not been established. And we already mentioned that in the modern medical microbiology sorts, our intestinal area, including small and large intestinal area, the bacterial community is our immune system. So that one has not been established. So they cannot combat for the honey if they contains Clostridium, even a very low concentration. Somehow even 10 cells could cause a problem. Why it was a floppy baby? Because the muscle relaxing. Why? Because this botulism toxin will be attacking acetylcholine. And acetylcholine is responsible for muscle contraction. So, if this guy is gone, the muscle you cannot contract it, then the muscle is always relaxed. So it looks like that. That's infant botulism. You remember this joke, and you will understand what is infant botulism. Okay, food botulism, we want to talk more about that. What is food botulism? It's related to one group of the foods, which is called canned foods. Okay, what is the canned foods? Canned foods, we also call it low acid food. What is the typical characteristics for low acid food? Is pH around 4.6, water activity about 0.85. This gives you a very favorable environment for endospore to grow. So we want to talk about that. Okay. Let's say I have a can. Okay. What we have? Some tomato. It could be some piece of tomato. Okay. Some piece of tomato. Some piece of like a pickled cucumber. And some water. And we add some salt there. Where those cucumber and the tomato originally come from? Same thing. from the soil. Once they coming from the soil, it could be contaminated with Clostridium. And then you could say, okay, why you not just wash it? And I want to tell you one thing, that tons of research has been showing, even you clean it, you sanitize it, the surface of those vegetables and the fresh produce, it is about 3 log bacteria there. It could be Clostridium. And we did the sampling before. You just grab a bag, even the packaged bag from Kruger, the ready-to-eat slides, you open it. I want to tell you one thing, you can test in the lab at least 100,000 cells there sometimes, at least 10,000 cells there. It's a lot of bacteria, it's not sterilized, which means even you wash it and process it does not mean there's no bacteria, there are tons of them. Okay, so that's something we wanted to know. Okay, so what we do? Why will cause a problem? Well, sometimes you have beans. Okay, you have beans. In the old time, lots of people to do is blanching. That means you boil 2 hours. Is this going to kill bacteria? Yes. They can kill vegetables. And inactivate enzyme. Because especially you study plant science, you know that there's lots of enzymes there. We don't want them to be damaged the quality, so we do the blanching, boiling 2 hours to kill the enzymes, kill vegetative cells. But how about the endospore? No, there's no effect for endospore. Endospore will survive, but actually will be dominant. Instead of survive, will be dominant. Because all the vegetative cells has been died, the endospore start to multiplication, it's dominant the whole can. So what people do, especially for those small processors, later on they come out a process called a pressure cooker. And the pressure cooker is actually a pretty nice facility for those small family-based processors. It's required 15 psi for pressure, 121 degrees Celsius for temperature, and at least 15 minutes for the processor. This will be very effective for killing the endospore. Now, how are we going to drop the pH? Remember, pH more than 4.6 is not good because endospore will grow. How we do? People add lemon juice, vinegar to decrease the pH below 4.6. Okay, very important in the West Virginia. In West Virginia area, we require pH less than 4.0. That's the only requirements in this local community for the canned food. The pH less than 4.0, then you tint it, you're good to go at farmers markets. Now, how do we know that canned food has been contaminated and not good? You see these type of convex, that's not good. You see some of the dents, that's not good. And you see some of the liquid extract, that's not good, which means the bacteria start to survive, start to grow. Okay, so that is the food organism. And you see the picture right there on the top, the canned food, the low acid food. See the pickles there, and the salt and salts there. It's preserve a little bit. Now I also want to mention that some people put salts in. Yes, that's good. Like 2%, 5% salt, 2 to 5% salt. That is also favorable to killing the bacteria. Some of the bacteria will be killed. However, halophiles will survive. And also, I want to tell you, lactic acid bacteria will survive. So what happened is, they will go through a fermentation process. That's why you pickle later on become acid. And also, the salt is also going to impact the softness, firmness of the product. So some people add salt. But I want to tell you one thing. Salt does not have much impact for killing endospores. Nothing. It's only for vegetative cells. And basically it's for fermentation, and it is also for the generate the quality of the canned food products. Okay? This is a pressure cooker. That's floppy baby, and this one is a room. Okay? So that's Clostridium botulism. Okay, Clostridium tetanus. Clostridium tetanus actually is very easy to understand. So we could just take out here and talk about Clostridium tetanus. Okay, Clostridium tetanus, number one. This should not be a problem in the United States because we have a vaccine, BTAD. We talked about D, diphtheria. We talked about P, Bordetella pertussis. For whooping cough, what is P? Clostridium tetanus. So in the United States, it's not a problem. There is basically the problem happens is people that don't have a vaccination and also coming from the cuts. Contaminated with the soil or decayed metal. And what's going to happen? The final symptom is lockjaw. Why? Because tetanus toxin attacking a chemical called glycine. And this glycine is responsible for muscle relaxing. If some neurotransmitter responsible for muscle relaxing is being blocked, your muscle is always contracted. This is like this. Why we can do that? Because the acetylcholine and the glycine is keep a balance. If one of them is blocked by the toxin, you either cannot do relaxing or you either completely contracted. Okay? That's something I want to mention. Third thing, this is actually a big risk for some developing countries in the older time during home delivery, home baby delivery. So it will generate a neonatal tetanus.